So the instrument displayed in front of you is called as improved Neuber counting chamber, or if you want to name it accurately, it's called as hemocytometer slide with improved Neuber counting chamber. Okay, this instrument that we nowadays use in the clinics, in the laboratory, this was first invented by a researcher named Kramer. Okay, in the year 1885. The prototype of this instrument was invented by Kramer, and later on, after few modifications and some improvements, it was invented by a person named Neuber. And giving due credit to the person who invented this present model, we name this instrument as hemocytometer slide with improved Neuber counting chamber, because Neuber did some modification in the counting areas. So this is a thick slide as compared to the glass slides we use for differential leukocyte count, okay, so, or any blood smear examination. So we tend to get a very thick smear generally, usually in those slides. But in this thick slide, or this glass slide or hemocytometer slide, if I'm using a cover slip, okay, placing it over here, now due to the placing of the cover slip, there will be exactly 1/10 millimeter of space in between the cover slip and the thick glass slide. Okay, so then we actually get a very uniform layer of cells, and we are going to get a very better result as compared to the thick glasses. Okay, so this slide is used to obtain very thin film of fluid. So this slide is used to it helps us to give or make a very thin film of fluid of any known fluid volume or blood, okay, containing the cellular components. So this is a hemocytometer slide. So obviously we do the examinations for counting the number of cells, whether it's an RBC, whether it's platelet or WBC, whatever. Okay, so let's learn about the parts of the instrument one by one. Okay, so from you are viewing from the top of the slide. Okay, so obviously this slide has a longitudinal edge or broad edge or long edge, and this is called as the narrow edge of the slide. Okay, there are the information or the legends given about the information about this slide, that is newer, improved. There are some optometric parameters are given. Okay, we don't need those. You keep your focus on the structures that is present over on the top. Okay, so coming from the sides, okay, these are called as the side platforms. Okay, side platforms of the cover of the Neuber slide. So these areas, this flattened area, if you're looking from the side, then you can see there is two ridges and there are three elevated parts. Okay, one is this, another is this, another is this. Okay, so when you are viewing from the side, you are getting three elevated areas and two on either side. So there are total of four ridges are there. Okay, so the side elevated raised area is called as the side elevated or raised platforms. Okay, adjacent to the side elevated platforms we have a gutter. Okay, these are called as your, so these two areas are called as the gutters or the side gutters or lateral gutters of the instrument. Okay, so this and these two are called as the gutters or the side gutters or lateral gutters. Okay, now inside you can see there is two elevated elevated areas or platforms. So these are called as your modes. Okay, these are called as modes, and in between the modes we have an H-shaped group. Okay, you can see there is a ridge these two ridges are there, which are making a communication in the center and dividing the whole structure into an H shape. Okay, so this is called as H-shaped groove or H-shaped gutter. Okay, or sometimes it is also called as the transverse gutter and horizontal and vertical, transverse or longitudinal. Sorry, this is the transverse and this is the longitudinal gutter. Okay, now in between the two transverse gutters we can see there are silvery shiny areas. Okay, if you are trying to reflect the sunlight, you can see two shiny central areas are there in between the two lateral gutters, transverse gutters. So you will get these two areas are called as elevated central platforms or Neubaur counting area. So these two areas are called as the central depressed platforms. Okay, central depressed platform having two shiny rolling areas. Okay, there. So inside this area we have some shiny silvery shiny rolling areas, which are called as the rolling areas or Neubaur counting area. Okay, so when this is as a whole, when we are looking from the top without placing any cover slip, this area is called as the area of the Neubaur counting area. Now if I am placing a cover slip, what happens? I told you the central area is a depressed platform, okay, as compared to that of the middle platform. Okay, so when we are placing a cover slip, what happens? The cover slip is standing on top of this raised platform and this raised platform, while the gutter, A-shaped gutter, and this central depressed platform or the Neubaur counting area will be at the little bit at a lower height. So at this particular point, the Neubaur counting area becomes into a chamber. Now chamber, you can think it to be like you just when you place a cover slip, it converts the floor into a three-dimensional structure. Okay, so then it becomes a Neubaur counting chamber. So it has been seen the distance between the cover slip to the central depressed platform. If I am going to measure in a very microscopic level, then you will be able to see it's approximately around 1 by 10 millimeter. So inside this space there will be a thin layer of blood volume, which will help us to determine the count very clearly and very accurately. So inside the central platform of the newest hemocytometer slide, you will get the grid, which looks like this. Okay, so I told you in a hemocytometer slide there is an A-shaped groove. Okay, this H-shaped groove divides the central depressed platform into two distinct areas. Now if you look closely under the microscope, you will get grids. Now each of these grids are present there. So in one hemocytometer slide we get two such grids. Okay, now let's learn about the individual parts of the grid or the hemocytometer grid with improved Neubaur counting area. And these grids are made of some squares, some bigger squares, some larger squares, some smaller squares. Okay, so each of the squares are used, and the areas of the grid are designated for counting the cellular components of the blood. That is the WBC, that is the white blood cell, RBC, the red blood cell, platelets, okay, and sometimes also the sperm counts in spatial laboratories. Okay, so these are the components that we are checking with the help of this hemocytometer slide. Now let's learn about the individual grid parts. Okay, so if I am measuring the length and breadth of this grid, so the length of the grid, so it's a square shaped grid. Okay, so the length and breadth both are 3 millimeter. Okay, which are divided into 3 equal segments, both horizontally and vertically. So what happens, this 3 millimeter is divided into 1 millimeter each part. So the length of this part is 1 millimeter, the length of this part is 1 millimeter, the length of this part is also 1 millimeter. Similarly goes over here, 1 millimeter, 1 millimeter, 1 millimeter. So let's keep our focus on this part of the Neubaeuer counting area. Okay, so this is called as the WBC counting area. Okay, this is called as the WBC counting area. So in the WBC counting area, you can see there are some smaller, some big boxes are there. Okay, big square boxes are there. In the very center of the grid, you can see there are some small grids are there. This is called as the RBC counting area. Okay, so here we do all our RBC counting. Okay, only this area that I'm showing you over here. Okay, so here the RBC, the platelet, the sperm count, even leukemic WBC count is done. Okay, so what we do, let's learn about the areas of individual components. Sometimes questions are framed from the areas of the individual squares of the grid. Supposedly the questions might they ask you what is the area of this square, the shaded area of this square of the Neubaeuer counting grid, or they ask you about the smallest square in the grid, in the RBC grid, what is the area of this. Okay, so you also have to know about the nomenclature of the different grids. Okay, so this as such, this square, the bigger squares on the four corners of the grid is called as the WBC large square. Okay, now these large squares are divided into 4 equal parts, both horizontally and vertically. So individual cells are also being formed which are a square shaped area. So this red shaped area, shaded area is called as WBC small square. Okay, so there are 16 such small square, 4 in this, 4 in this, so 4 by 4 there are 16 such small squares. Okay, 16 small squares are there, small WBC squares. So in the Neubaeuer counting grid we have 16 4 times. So there is 1 2 3 4, there are 4 large squares, so 16 into 4, 4 5 6, so we have total of 64 WBC small squares. Okay, so what is the area of one WBC large square? Okay, simultaneously your next question will be what is the area of one WBC small square? Okay, so let's calculate the large square first. Okay, so I've told you the whole Neubaeuer counting grid's length is 3, which is divided into 3 equal parts. So we have the length of the large square is 1 millimeter. Similarly, the grid is also divided 3 times in the vertically also. So the length of the large WBC square is also this. So therefore the area of one WBC large square is equals to length into breadth, that is 1 times 1 millimeter square, that is 1 millimeter square. Okay, now if you want to convert that into the volume, what will be the volume? Volume of one WBC large square is area times the height. Now I've told you when we place the Neubaeuer counting area horizontally,
and we place a cover slip on top, what happens? There is a potential space in between the cover slip, okay, in between the Neubaeuer counting grid and your cover slip. That's height is 1 by 10 millimeter, okay, 10 by millimeter. So that is nothing but the height of our WC square also, or the RBC square also. The whole grid has a height of 1 by 10. So let's multiply in this area. 1 millimeter square is the area and height is 1 by 10 millimeter. Okay, so now the volume of this whole chamber becomes, this one single RBC WBC large square becomes 1 by 10 millimeter cube. Now let's count let's find out the area of RBC small square. Okay, this area, the shaded area, this is called as RBC small square. So now the length, I told you the length of this RBC WBC large square is 1 millimeter. Okay, now look closely. This RBC length is also again divided into 4 equal parts. Okay, so when 1 millimeter is divided in 4 equal parts, the value becomes 1 millimeter of each part of the segment of the, or each segment of the length of the small square, small WBC square. Okay, so we can say that this length, okay, is, okay, let me erase all the parts. Okay, so this part is 1 by 4 millimeter. Now similarly, look closely over here. This is 1 millimeter, right, which you can see again been divided into 4 equal parts, right? So individually the 1 millimeter is divided into, the whole 1 millimeter is divided into 1 by 4. This part is 1 by 4 of a millimeter. Okay, so we can say that the breadth, okay, of this WBC small square is 1 by 4 millimeter. Okay, so therefore area of 1 WBC small square is equals to length into breadth. Length is 1 by 4, breadth is 1 by 4, and we are getting the unit in square. So we have an area of 1 by 16 millimeter square. So done. Now similarly, if you want to calculate the volume of 1 WBC small square, okay, what will be that? So 1 WBC small square, okay, volume will be area into height. Now we know when we place a cover slip, there is a space of 1 by 10 millimeter between the cover slip and the Neubaeuer counting grid. So that will be the height of the chamber. So then area is 1 by 16 coming from this, and height is 1 by 10, and the units are in millimeter cube. So we get the volume of 1 WBC small square as 160 millimeter cube. Okay, so how many WBC squares are there? How many small squares are there? So in individual WBC large square, we can see there are 16 squares, small squares. So there is 16 small square over here, there is 16 small square over here, there is 16 small square over here, so 64. So therefore the total volume of 64 WBC small square is 160 into 64. So we calculate the value. So the value comes out to be 0.4 millimeter cube. Okay, now that was the WBC small square. Okay, So this is the WBC small square. The important point is what is the area. Area of what WBC small square is 1 by 16 millimeter square, and the area of WBC large square is 1 millimeter square. So these 2 points are to be noted. Okay, next move on to the RBC counting grid. Okay, so I told you this whole segment, this whole area at the very center of the Neubaeuer counting grid is called as, this whole area is called as the RBC counting grid. Okay, so under this counting grid, if you look closely under the high power microscope, then you will see that individually these grids are divided into some triple lines. Okay, so basically in this picture it's not showing you, but the thick lines are actually triple lines. Okay, so individually all these are triple lines, these are triple lines. So everywhere these triple lines are intersecting through and throughout the middle counting RBC counting area. Okay, so wherever it is dividing or intersecting the lines horizontally or vertically, it has been intersected by the triple lines. Okay, so that's the modification of the improved Neubaeuer slide. Now you look closely, this, as I told you at the beginning also, the Neubaeuer counting grid's length is 3 millimeter, which is divided into 3 equal parts. So individual length of the part is 1 millimeter. So that is same for this whole square. Okay, that's also 1 millimeter. Similarly, it goes in these areas also. In the vertically, if you measure, it's also the same 1 millimeter. Okay, here also is the square. So square is divided into 3 equal parts. You will get individual parts to be the equal size only. Okay, now let's keep our focus on the squares. Now in the WBC, I told you there are 2 squares. One is a large square, okay, and another is the small square. Okay, but in this case, in the RBC counting, when we name the squares, we name based upon three values. Okay, so there are 3 types of squares you can see. Okay, one is this whole counting grid. Okay, this whole counting grid is called as RBC large square. Okay, now there are triple lines. I told you the triple lines are intersecting. Okay, now because of the intersection, what happens, the large squares are divided into a square which are of medium size. So this is called as RBC medium square. Okay, now if you look closely, these triple lines are making the small squares, medium squares. Now even the medium squares are being divided into 4 equal parts. Let's keep our focus in this square. So it looks like a WBC square, kind of a square having a 16 4 by 4, that is total 16 squares are there inside this medium square. So this is the medium square, and inside this medium square, if I'm enlarging this square, if I'm trying to enlarge this square and trying to draw, there will be triple lines surrounding this block. Okay, triple lines will be there everywhere. Okay, intersecting, but this RBC medium square are again divided into 4 equal parts by a single line. Okay, so then what happens, this square, okay, that's the small pinch of square that you can see from the whole grid. This is this. Okay, it is representing this. So what will be the length and breadth of this one? Okay, and what is the name of this? So this is called as the RBC small square. Okay, so these are the nomenclature. Let's learn about the dimensions of the squares and the areas. Okay, so let's start our counting, or let's start our area calculation from the RBC large square. Okay, that means this square. So I told you a 3 millimeter grid is divided into 3 equal parts. So the length of this grid is 1 millimeter. The breadth of this grid is also 1 millimeter. Okay, so the area of this grid is length into breadth, that is 1 by 1 millimeter square. You get the value to be 1 millimeter square. Okay, similarly, like the RBC WBC large square, you will get the volume as 1 by 10 millimeter cube. Okay, I'm not going to go into the volume part, just let us calculate the area part. Now let's calculate the RBC medium square values. Okay, so what will be the length? First of all, let's find out the length and breadth of this small grid, that is this grid. Okay, now I've told you this grid. Okay, it has a value of this length. This has a value of 1 millimeter. Okay, now if you can see, the triple lines are dividing this grid length into 5 equal parts. Okay, 1 2 3 4 5. So this 1 millimeter section is divided into 5 parts. So what will be the length of this square, this particular square? Okay, or this particular square? Okay, this is 1 divided by 5. Okay, it's divided into 5 equal parts. Okay, so that will be the length. Similarly, here also you can see this is 1 millimeter. Okay, which are divided by the triple lines into 5 equal parts. So 1 millimeter is divided into 5. So here also the breadth will be the same only, that is 1 by 5 millimeter. Okay, now we have found the length and breadth of the RBC medium square. So area. Okay, so what will be the area? So area will be length into breadth. So 1 by 5 into 1 by 5 millimeter square. So you should get the values of 1 by 25 millimeter square. Okay, now let's calculate the smallest square. Now what will be the area, length and breadth of the RBC small square? Now we will have to calculate the parameters of the RBC small square. Okay, what will be the length, what will be the breadth? Let's find out. So up till now we found out that the length of RBC medium square is 1 by 5 and breadth of the RBC medium square is 1 by 5. That means, let me enlarge this value. Okay, so this was 1 by 5. Okay, so you can see the triple lines are dividing the square's length into 5 equal parts. Okay, sorry, 4 equal parts. So what will be the value of this individual cell, the individual length? That is 1 by 5 is divided into 4 times, that is 1 by 20. Similarly, here also you can see, you know, the length of this RBC medium square we know is 1 by 5. Okay, so which is divided into by a single straight line, which is divided into 4 equal parts. So what will be the value? 1 by 5, the length is divided into 4 equal parts. So therefore the value of the breadth will be 1 by 20. Okay, let's go back to our calculation. So we have found that the length of the RBC small square is 1 by 20 millimeter. Okay, and breadth is also 1 by 20 millimeter. Okay, so therefore the area will be length into breadth, that is 1 by 20 times 1 by 20, okay, millimeter square. So therefore you should get the value as 400 millimeter square. Now what will be the value of all the RBCs? That's a humongous thing to count under microscope. So while we are counting with the help of a microscope, we don't count all the cells. Whenever we are counting, we are actually keeping our focus on the small square only. Okay, we're so focusing on the small square. So how many such squares will be there? So individually we have 16, okay, square in individuals RBC. Medium square we have 16 small square. Okay, so in one RBC medium square we have 16 RBC small square. Okay, so how many such medium squares are there? I told you the triple lines are divided into 25. So 1 2 3 4 5, 1 2 3 4 5, so there will be 25 RBC medium squares. So 25 RBC medium squares will have 16 times 25, so we get the value of 400. So there are 400 small squares. So while you are conducting an experiment of RBC count supposedly, and we are going to count, do you think it's possible to count 400? Because the counts will be in a millions, so it will be humanly impossible. So hematologists did what? They did some strategic calculation. Okay, if supposedly you are charging the slide with the help of a blood volume. Okay, charging means filling the Neubauer counting chamber with the blood accurately and proportionately. Okay, when it is happening, what happens? The RBCs are proportionally distributed. Supposedly you are counting the four corners, strategically taking the four edges of the volume distribution in the RBC grid, and also taking the very central square, medium square, then it's little bit possible to count, because now you are counting not 400 but 16 times 4, that is 80 RBC small square. So from 400 you can just count 80, so that's humanly possible. So in actual practice, if you want to do the RBC counting or the platelet RBC counting especially, you have to count the 5 RBC medium squares and all the small squares that are present in the RBCs, 5 peripheral corners and 1 central RBC medium square, and all the small squares that are present inside these medium squares. So therefore what will happen to the area? So actually we are counting this much wall, this much area. Okay, so this much area of count we are getting. So 80 times 80 by 100, sorry, 1 by 400. Okay, so this is the area of the Neubauer counting grid. Okay, while we are doing the platelet count, we take the consideration of all the 25 small squares. We don't fix our counting to just 5 RBC medium squares, we take all the 400 small squares. So that's it for the RBC calculation of the RBC counting grid and the WBC counting grid.